Today we are starting with the bacterial electron transport. So in prokaryotes, usually the electron transport chain is very complicated. First you should know what is electron transport chain. Until now we have seen that the EMP pathway, then later on the citric acid cycle or the Krebs cycle generates NADH and FADH2. So these NADH and FADH2, it provides electrons or it transfers electron from these molecules and they themselves get oxidized. So what happens when the NADH gives the electrons to the another electron acceptor molecule, it becomes NAD positive. Similarly, FADH2, when it donates electron or transfers electron, it becomes FAD positive. Okay. So these electrons now enter through different electron accepting molecules. So you will see what are the electron donors, then how they are donating the electrons to several different electron acceptors. So why they are donating electrons and why it is important? It is so because that in order to acquire or in order to gain energy in the form of ATP. See, if a bacterial cell or any cell undergoes EMP pathway or glycolysis, it generates only 2 ATP molecules. So net gain of ATP is 2 in EMP pathway, as well as in citric acid cycle usually there is gain of only that is 4 ATP, but there is gain of 6 NADH molecules, that is in the form of reduced NADH molecules, as well as 2 FADH2. So obviously from one NADH, because of transfer of 2 electrons to a cascade in the electron transport chain or electron transport system, one NADH molecule can give you 2.5 or 3 ATP molecules. So what happens when you have 6 NADH molecules generated via that Krebs cycle and EMP pathway, then you will have near about 18 ATP molecules which cannot be given only either by EMP pathway or by that Krebs cycle. So it is important, in order to have many more ATP energy molecules, the bacteria as well as the eukaryotic cell undergoes electron transport chain. So what is the advantage or significance of electron transport chain? Electron transport chain usually is necessary only to have many number of ATP molecules. Okay. Now we will come to bacterial electron transport chain and then we will see the electron transport chain in eukaryotic mitochondrial cell. So first we are going to see the bacterial electron transport chain. So in prokaryotes the situation is complicated because there are several different electron donors and several electron acceptors. So the generalized electron transport chain in bacteria is from donor it can be a hydrogenase, so NADH dehydrogenase, or it can be a quinone, the second donor. So from quinone it can transfer the electrons to the BC1, and BC1 is also a complex which is acting as a donor, and then it can transfer its electron to the cytochrome. Later on from cytochrome or from this it is undergoing a transfer of electrons to the oxidases, or reductases. See, when the ETC takes place in aerobic condition or aerobic condition, then the last electron acceptor is oxygen. So here oxidases enzyme is undergoing the oxidation process in which O2 is converted to water molecule. So the last electron acceptor when it is using oxidases is a oxygen molecule and the proton molecules which forms a water molecule. But when it is undergoing a anaerobic condition, then you will see that the reductase is undergoing the process of conversion of different other molecules like ammonia, nitrate, nitrite, ferrous, etc. They are acting as a last electron donor and then they are converted to respective that is reduced molecules. So here the electron acceptor, terminal electron acceptors are either the nitrate or the ammonia, or it can be a, hydrogen sulfide, etc. So different forms are usually found in the microorganisms. So you have a generalized idea now how the electrons are donated and what are the terminal electron acceptors. So these electrons, it enters the chain at 3 levels. So what are the 3 levels? One is dehydrogenase level. So another one at the level of the quinone pool or at the level of mobile cytochrome carrier. So these levels, they usually correspond to successively more positive redox potential or to successively decreased potential. There is a difference in between the that is the PO means oxygen as well as the proton. So proton and oxygen potential difference is there. Because of this, the electrons are flown or there is a flow of electron via Gibbs free energy chain, so that overall redox reaction takes place in which the electrons flow from donor to the acceptor. So individual bacteria usually it uses the multiple. So when you observe a bacterial cell, that bacterial cell typically it can use multiple electron transport chains. So they can use 4 to 5 electron transport chains simultaneously, and they can have number of different oxidases and reductases and a number of different electron acceptors too. So for example, in E. coli, it uses 2 different NADH dehydrogenases and 2 different quinol oxidases. So total it uses 4 different electron transport chain, and these 4 different electron transport chains operates simultaneously. So another common feature of electron transport chain is the presence of a transport, that is proton pump, to create a electrochemical gradient over the membrane. This I will explain you. So it is very similar to the mitochondria. You know that bacterial cell possesses a cell wall, then you have a cytoplasmic membrane, and inside you call it as a cytoplasm, and the outside of the cytoplasmic membrane you have a periplasmic space. So the protons are usually pumped from the inside, that is from the cytoplasm to the periplasmic space. So how it is pumped out? Usually the protons are pumped out, and these pumped protons usually they form a proton gradient, and because of the formation of large number of proton concentration in the periplasmic space, it drives the synthesis of ATP formation. So electron donors usually in present day biosphere, the most common electron donors, what are those? They can be organotrophs like animals, fungi, protists, or they can be a phototrophs like plants and algae. Some prokaryotic organisms, they also use inorganic matter. So organotrophs and phototrophs, they use the organic matters or the organic molecules as the electron donors. But if the prokaryotic cells uses inorganic matter as an energy source, then they are usually referred as lithotrophs, and these inorganic electron donors include hydrogen, carbon monoxide, nitrite, then ammonia, sulfur, sulfide, manganese oxide and ferrous ion. So lithotrophic organisms, usually they donate electrons from inorganic energy sources like hydrogen, carbon dioxide, nitrite, ammonia, sulfur, sulfide, manganese oxide, etc. So these electron donors, they are of particular interest because of them we can know the evolution or the evolution of that particular microorganisms. Now, in the electron transport chain of bacteria, you have different complexes. So first we will see the complex 1 and complex 2. So bacteria, they use number of different electron donors. When organic matter is the energy source, the donor may be NADH or succinate, in which case electrons enter the electron transport chain via NADH dehydrogenase. So this is your complex 1, which is also referred as NADH dehydrogenase, or you can call it as the another complex, that is complex 2, which is succinate dehydrogenase. So these are the two complexes which are usually donating the electrons. Also you have another dehydrogenases which possesses different energy source like formate dehydrogenase, lactate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, hydrogen dehydrogenase in the electron trans. So mostly, that is the complex 1 and the complex 2 transfers its electrons to the another. The second, where it transfers the electron now, this NADH dehydrogenase and succinate dehydrogenase, it transfers its electron to the quinone carriers. So quinone carriers, now quinones, what are quinones? Usually they are the mobile lipid soluble carriers, and they shuttle the electrons or protons between the large relatively immobile macromolecules like complex 1 and complex 2, complex 4 and complex 3. So usually what happens, this ubiquinone or coenzyme Q, they are the same as that of the mitochondria. So mostly the bacteria, they use ubiquinone, and if they use menaquinone, that is vitamin K2, they can also use the menaquinone. So either they can use ubiquinone or they can also use the related quinones, which is called as the menaquinone. So the use of different quinones is due to slightly altered redox potential. So what happens when there is a difference between the potential gradient or electrochemical potential because of the transferring of the protons from inner cytoplasmic content to the periplasmic space. Because of that, the electrochemical potential changes, and because of this, what happens, there is a difference in the redox potential. So accordingly, the electrons are transferred from one system to the another. Now what happens, because of the transfer of electrons, the complexes gets charged, and when they get charged, they usually flow or transfer their electron in order to stabilize themselves, or in order to acquire their equilibrium, or in order to acquire their own ground state. So when they are again transferring the electrons, usually what happens, they come to their normal position. So during this, what happens, when it is in the charged condition, they pump the protons from inside the cytoplasm to the outside, and this creates a proton gradient across the membrane. So because of the proton gradient, usually what happens when the two electrons are transferred from NADH, near about 16 protons are getting outside from the cytoplasm to the periplasmic space. So this thing you have to very clearly keep it in mind, that because of flow of two electrons from NADH to the in the transport system, that is electron transport chain, you will have 16 protons moved outside the cytoplasm into the periplasmic space. So because of that, you will have a proton gradient across the membrane. Now usually the protons are physically moved across the membrane, and this is usually seen in mitochondrial complex 1 and complex 4. This tomorrow we will see what are the complexes and what are the names or the designations of these complexes. So similarly, it is moving the protons from the cytoplasm to the periplasmic space or to the periplasm. Some dehydrogenase, they are the proton pumps and others are not. So most oxidases and reductase are proton pumps, but some are not. So what are those proton pumps? Who is pumping the proton from the cytoplasm to outside in the periplasm? It is the BC1, which is a proton pump found in many, but in not all bacteria, and it is also not found in E. coli. As the name implies, BC1 is similar to mitochondrial BC1, which is called as the complex 3. Now the electron is moving. So it is moving to the cytochromes. So these cytochromes, you have cytochrome C in bacteria. So these cytochromes, usually what are those cytochromes? So cytochromes are the colored pigment and they contain the iron. So in very different environments you can find cytochromes in many variations. So some cytochromes, they are water soluble carriers that shuttle electrons to and from large immobile macromolecule structures embedded in the membrane. The mobile cytochrome electron carrier in mitochondria is the cytochrome C, but in bacteria it uses the AA3, etc. So other cytochromes are found within the macromolecules such as complex 3 and complex 4. So they are functioning as the electron carrier. They transfer the electron from one level to the another level. So electrons, they enter the electron chain at the level of mobile cytochrome or quinone carrier, and then they transfer the electrons at the another level, where electrons enter at the redox level greater than NADH. So it operates in a cascade or in a stepwise steps in which the electrons are transferred. Now the terminal oxidases and reductase. So when bacteria is growing in aerobic environment, usually the terminal electron acceptor is oxygen, which is reduced to water by enzyme oxidase. But when bacteria is growing in anaerobic conditions, then the terminal electron acceptor is reduced by the enzyme called the reductase. So in mitochondria also you will see the terminal membrane, that is complex 4, is cytochrome oxidase. So usually you will find that there are different terminal oxidases. So in E. coli, a facultative anaerobe, it has cytochrome oxidase or BC1 complex. So this is your terminal electron acceptor in which it reduces the oxygen to water. But what happens when it is in anaerobic condition? So usually the bacterial complex 4 can be split into two classes. One is cytochrome BC1 and the another class is AA3. So AA3 is a heme AA3 class 1 terminal oxidases, which is more efficient than the class 2 terminal oxidases. Now terminal oxidase, class 2 terminal oxidases are the quinol oxidases, and class 1 oxidases, they are the cytochrome oxidases. So cytochrome oxidases, they use oxygen as the terminal electron acceptor, and the quinol oxidases, they use variety of terminal electron acceptors. In anaerobic conditions, what happens? The reductase enzyme individualized into the terminal acceptor, they use fumarate reductase or nitrate reductase or nitrite reductase or trimethylamine and oxide reductase, anything. So, this in anaerobic conditions, it is the reductase which is the terminal electron acceptors. So, in most cases these oxidases or reductases, they are inducible, they are synthesized by the organism as indeed when needed and when in response to specific environmental conditions. So, usually what happens in aerobic conditions, oxidases are synthesized to carry out the electron transport chain, but in anaerobic situations, usually the reductases usually are synthesized or induced to undergo electron transport chain. So, until now we have seen the electron acceptors, what are the electron acceptors and electron donors. Now, we will see the diagrammatic representation of, that is, bacterial ETC. So, you know that NADH is, or NADH dehydrogenase is donating the electrons, and these electrons which are donated are transferred to the ubiquinone. So, this is QH2 ubiquinone. Now, you have two different environmental conditions. One is high aeration when the bacteria is growing in the log phase, and the another condition is low aeration, that is the anaerobic condition, usually when the bacteria is growing in the stationary phase. So, this is your cytoplasm. In the cytoplasm, you have a proton, here protons are there. So, because see NADH plus H plus, so this is the proton. So, whenever there is NADH, you have the large amount of protons. So, these electrons are flown then to the FAD. FAD is the another electron donor, and this FAD molecule transfers electrons to the FES centers. You have 4 FES centers, and from the FES centers the electrons are transferred to the ubiquinone. So, to ubiquinone it is transferred in both the conditions. So, here also you have QH2, here also you have QH2. So, when QH2 is, now what happens at low aeration or in stationary phase, this ubiquinone transfers electron to the cytochrome B558. So, this is your cytochrome B558, and because of this what happens, the electrons are then passed to the cytochrome, that is your B96. Okay. So, when it is doing so, what happens? The electrons from the cytoplasm is moving outside. See here, 2 protons are moved outside in the periplasmic space. Similarly, when it is transferring the electrons, 2 electrons, then in high aeration or in the log phase, it is first transferring its electrons to cytochrome B562. And then it is transferring these 2 electrons to the cytochrome O. So, in high aeration or in the aerobic condition, it is transferring to the cytochrome O, and when the anaerobic condition prevails or when there is low oxygen, then usually it is transferring to the D. So, usually it is transferring to the D. This is your D. So, cytochrome this is cytochrome O and this is cytochrome OC, or you can call it as cytochrome D. So, this is, so because of that you will see that there is a 2 branches. One branch is called as the cytochrome D branch and the another one is called as the cytochrome O branch. So, what happens? NADH is the electron donor, ubiquinone is Connecting link between the dehydrogenase, NADH dehydrogenase with 2 terminal oxidase of the 2 branches, that is cytochrome D branch shown in 1 and cytochrome B O branch, this is shown in 2. Okay, so this is your 2 and this is your 1. So what happens because of this? What happens? So in aerobic condition, the oxidases reduces the oxygen to water, and it is utilizing which cycle, that is the cytochrome O. But in anaerobic condition, usually it is following the cytochrome. The last electron acceptor is the cytochrome D. Okay, and this cytochrome D is shows very high affinity of oxygen, and it always operates at the low oxygen level or low aeration. So usually in the stationary phase of growth. So this is how the electron transport chain works in the bacterial cell. Now, what is photophosphorylation? Now in electron transport chain, we have seen only the transfer of electrons from one electron donor to the electron acceptors. So you have many 4 different complexes. Then you have seen the transfer of electron from complex 1 to the complex to the ubiquinon, and then to the from the ubiquinon to the cytochrome that is cytochrome C1, and then to the last electron acceptor, that is the oxygen or the cytochrome oxidase. So in this way we have seen the electron transport chain. But what is phosphorylation or oxidative phosphorylation? It means that when the electron transport chain is generating a proton gradient in the periplasmic space, you will have higher concentration of protons in the periplasmic space. Now these protons are again moving inside the okay. So if this is your bacterial cell, then from bacterial cytoplasmic membrane, this is your cytoplasm. From inside to the cytoplasm, the proton is pumped outside into the periplasmic space. So here you have a pool of protons outside in the periplasmic space. So when you have a proton in the periplasmic space, the protons are again entering into the cytoplasm. So while doing so, while entering, the ATP synthase enzyme is taking advantage of this proton motive force and is phosphorylating the ADP into ATP molecule, and because of this what happens, you will have the ATP formation or ATP generation. That is what is called as the oxidative phosphorylation. Hope you have understood this. Tomorrow once again we will take it with the help of the PPT, and you will be able to understand better. Thank you.